Hello everybody, welcome to Deeksha Online. So we were discussing about the chapter Reproductive Health. So moving on to the next lecture of this chapter, that is lecture 3 of Reproductive Health. What was the roadmap or the chapter overview? Now we have already discussed about the reproductive health, what are the problems and how the nation wants to tackle those problems, that is strategies, correct? Then we also learned about population explosion and how to stabilize the population, and one of the means is by advocating the birth control options. Then we discussed about something called medical termination of pregnancy, that is MTP. And in this particular lecture, we will be discussing about sexually transmitted diseases or sexually transmitted infections, which are abbreviated as STI. And we will be discussing about the inability of couple to produce children, which is referred to as infertility. Okay. Now, before we get into today's lecture, as our routine discussion, we will go through a quick review, a quick recap of what has been done till today. Now, in the previous lecture, we had started discussing the methods of contraception. Now, in the methods of contraception, we had learnt about the barrier methods. Then we had learnt about the pharmacological or the chemical methods. The chemical methods are in the form of oral pills, oral contraceptives, or in the form of injectables and implants, or in the form of spermicidal creams or jellies or foams, isn't it? And then we had discussed about the surgical methods. In surgical methods, also called sterilization, we had called them as terminal methods. Vasectomy in case of male and tubectomy in case of female. And then we had discussed about voluntary termination of pregnancy before full term, intentional termination of pregnancy, which is called medical termination of pregnancy, where we had discussed some of the dangers, that is misuse of medical termination of pregnancy, and why did the government legalize MTP? Is it necessary to legalize MTP? What are the reasons why it is considered necessary? Okay. So with this review, we shall progress on to this particular lecture that I am dealing with right now. We will start off with sexually transmitted infections. Now, sexually transmitted infections are those which are transmitted through sexual intercourse or coitus. They are also referred to as the venereal diseases (VD) or reproductive tract infections. So please remember, all these are one marker questions. They will ask you to abbreviate, to expand the abbreviation STI. So STI stands for sexually transmitted infection, VD stands for venereal diseases, and RTI stands for reproductive tract infections. There are a lot of different types of sexually transmitted infections. To mention a few, we have gonorrhea, syphilis, genital herpes, chlamydiasis, genital warts, trichomoniasis, hepatitis B, and AIDS. So you can see there are so many diseases which are transmitted through sexual contact with an infected person or sexual intercourse with an infected person. So all these diseases are collectively referred to as sexually transmitted infections. Okay. There are a set of diseases, especially these three diseases, that is caused by mostly viruses: hepatitis B, genital herpes, and HIV. Why are these three diseases very dangerous? Because other than all these three diseases, there is cure if sexually transmitted infection is detected early and it is treated properly. That means these three diseases do not have a cure. So there is no cure for these three diseases. Now, all of you don't think that just because we say it is no cure, it is not a death sentence to the person, isn't it? Our general tendency is to believe that if a person is HIV positive, that means the person is doomed for his life is over, he is not going to live anymore. But it has been identified that of course diseases like hepatitis B can become extremely severe and it can be fatal, but there are people who have been surviving for a very long time with diseases such as genital herpes, and even HIV positive patients have been living very happily, fulfilling all their dreams. So when I say no cure, it doesn't mean that it is a death sentence. Please remember, it is manageable. If a person takes proper care of his personal hygiene, his lifestyle, his diet, then of course he can prolong his life. He can live a happy and content, and he can fulfill all his dreams. Okay, so there is a lot of misconception also associated with sexually transmitted infections, and that's why here again the importance of creating public awareness comes into picture. So remember, they may ask this as a 3 marker if they ask you to name all the diseases, or they may ask you to give an example of one of the sexually transmitted infections which do not have a cure. So you should remember the 3 diseases are hepatitis B, genital herpes, and HIV. Okay? Now, what are the early symptoms of sexually transmitted infection? It is very easy to neglect, it is very easy to overlook the early symptoms of sexually transmitted infection, and then it becomes a fully blown infection when you cannot control it. When it can lead to, ultimately, it can prove to be fatal and lead to death. So, let us go through some of the early symptoms, early signs and symptoms of sexually transmitted infections. Generally, there will be itching, mostly in the genital region of a person infected with STI. There may be a fluid discharge from the genital organs. There will be slight pain, and a sudden swelling may appear in the underarm, in the groin region. So, swelling of lymph node may be there, mostly in the groin or in the genital region. So these are sometimes the danger of these symptoms is that most of them do not take it seriously. They may consider it to be a routine illness, and they may just not even seek any professional advice. They may not seek any consultation from a specialist doctor. Okay, so remember, the early symptoms include any kind of a swelling, pain, discharge, and itching. And when I say swelling, it is mostly in the genital region. Another thing of concern when we talk about sexually transmitted infection is research has found that infected females may often remain asymptomatic for a very long period of time, meaning they may carry the pathogen in their body, in their system. They may have the bacteria or virus in their system, but they may not show any symptoms at all, isn't it? So if it remains asymptomatic, then it may remain undetected for a very long time. Now, why these early symptoms are a concern is because you all know about the social stigma associated with sexually transmitted infection. So, mostly, what does the person do? The person does not take these symptoms seriously. The person just overlooks them because he is also scared to go see a doctor for the fear of being labeled, isn't it? For the fear of the social stigma attached to the sexually transmitted infection. Once again, this is where awareness comes into picture. People need to be provided with proper awareness that even if they have the mildest of symptoms, they have to seek professional support, they have to get checked by a doctor, and so that if they are detected with the disease, then proper care and proper treatment can be given before it blows into a full infection, taking the life of the person. Okay, so they may ask you about the early signs and symptoms alone as a 2 mark question. So you should remember what are some of the early signs and symptoms of sexually transmitted infections. Okay. Then, what are the long term complications? Now, this also is a very important question in your board exam. Long term complications of sexually transmitted infections. Now, why does it become long term? I was telling you that if they do not take the early symptoms seriously, then obviously the disease is going to progress and it will express in the body in the form of long term complications. Mostly, when it reaches this particular point, medical intervention will be less effective and it can prove fatal to the person. So, pelvic inflammatory diseases, meaning the pelvic cavity will get infected, because pelvic cavity is where the reproductive organs are present, isn't it? The cervical canal, the vaginal tube, and then you have the uterus, the fallopian tube. Even in male, the vas deferens, the ejaculatory duct, the urethra, everything lies in the pelvic cavity. So, if the disease starts progressing, then from the reproductive tract, the pathogen may spread to the pelvic cavity. It may start attacking, it may start causing infection in all the internal organs of the pelvic cavity. This is called pelvic inflammatory disease. Then, abortion. It may lead to infertility. The woman may not be able to deliver or produce children. That is because time and again the woman may suffer from miscarriage or abortion. Or if she gives birth to the child has already died within the womb of the mother. This is called stillbirth. So abortion and stillbirth. Ectopic pregnancy. Now this is an important term all of you should remember. Do you recall where normally does the embryo get implanted inside the mother's body? Yes. Normally, the embryo implants into the uterine endometrium, but in some cases, it may get implanted, say, in the fallopian tube. So, ectopic means implantation of the embryo elsewhere other than the uterine endometrium. Such a pregnancy cannot be sustained. The embryo cannot grow further. It may cause serious medical complications in the mother as well, and such an embryo might have to be aborted, okay, medically terminated by carrying out MTP. So remember, if the embryo gets implanted elsewhere other than the uterine endometrium, then it is referred to as ectopic pregnancy. All this leads to infertility because the couple are unable to bear children because of all these complications. And if the sexually transmitted infection is not treated, it can also cause cancer of the reproductive tract. Isn't it? The cells lining the reproductive tract or the reproductive accessory ducts and the organs are so badly affected by these infectious agents that ultimately the cells may become cancerous. Mostly there are certain viruses, I think you must have heard, there are viruses called oncoviruses which are capable of causing cancer in the reproductive tract. Okay, for example, there is a virus called the human papilloma virus, which is a sexually transmitted infection, and it is capable of transforming the cells of the reproductive tract from normal cells into cancerous cells. So cancer is also a long-term complication of sexually transmitted infection. Okay. So we discussed what now? Two major things we discussed. First, we discussed the early signs and symptoms of sexually transmitted infection. Then we discussed about the long-term complications of sexually transmitted infections. Generally, from research and from statistics, it has been identified that people among the age group of 15 to 24 are more prone to suffering from sexually transmitted infections. So, there is higher incidences of sexually transmitted infections among people in the age group of 15 to 24. Now, there are a set of 3 principles one must follow. So remember, there are a set of 3 principles, or we can call them as modes of prevention, or we can call them as prophylactic measures. So if a person follows these 3 principles, it will act as preventive measure. It will protect the person from contracting sexually transmitted infections. So the question may be, they may say, what are the 3 principles one must follow in order to be free of sexually transmitted infection, or what are the 3 modes of prevention of sexually transmitted infection? The first mode of prevention, or the first principle, is avoiding sexual contact with unknown and multiple partners because they may be carriers of the infection, isn't it? So first one is avoiding sexual contact with unknown or multiple partners. The second one we had discussed in the birth control options, that condom does offer protection against sexually transmitted infection. So use of condom during sexual intercourse or coitus can reduce the chances of contracting sexually transmitted infections. Then the last one is extremely important. In case of doubt, one should see a qualified doctor. Now, this is done so that the person can get early detected in case the person has the sexually transmitted infection in the early stages. It can be detected and he can get complete treatment for the same if the person is diagnosed with STD. This is where the importance of what I was telling you, awareness, and to overcome the stigma, the social stigma attached to sexually transmitted infection becomes very important, because it should allow a person to seek qualified support in order to get detected at an early stage, and if he is detected, he will undergo complete treatment. Is that clear to all of you? So the three major principles or the modes of prevention of sexually transmitted infection. A very important 3-mark question, because it is a very basic answer. You can expect such very basic questions also in your board exam, which is very easily scorable. Of course, don't think it is very basic and don't try to write your own stories. Please use the terminology that is used here, which is as per your NCERT textbook. Is that clear? Okay, so now we have completed the sexually transmitted infection. Now let us move on to the next aspect in this chapter, which is referred to as infertility. This is the last section in this lecture as well as in this chapter, that is reproductive health. How do you define infertility? Inability to conceive. Conceive means fertilization. Fertilization between the sperm and the egg is not happening. Inability to conceive or produce children. So the couple are unable to conceive or they are unable to produce children even after 2 years of unprotected sexual cohabitation. So remember that even after 2 years of attempts towards conception, if they do not conceive or if they are unable to produce children, we call them as we call the couple as infertile. So here I would like to add a point here: unprotected sexual cohabitation. Okay, so inability to conceive or produce children even after 2 years of unprotected sexual cohabitation is referred to as infertility. Let us discuss some of the reasons for infertility. First, we will discuss about the reasons in case of male, and then we will go on to the female. In male, we had discussed something called cryptorchidism. Now, normally in the developing fetus, by the 7th month, the testis will descend down the abdomen and it will enter into the scrotum. In some children, they are born with a testis in their abdomen. There is no testis in the scrotum. So we call this condition as undescended testis. Okay, so undescended testis or cryptorchidism is referred to as is one of the causes of infertility in case of males. Then, if undescended, we had also discussed that undescended testis can be treated by using a corrective surgery, but of course, if it is not done, then the male will become infertile in the later stages of his reproductive life. Okay. Then, alcoholism inhibits spermatogenesis. You can consider alcoholism as a psychological factor, isn't it? There may be numerous psychological reasons as to why a person may become an addict towards alcoholism, and it has a direct link with the reduced sperm count, and that may make the male infertile. Now, another surprising thing is sometimes there may be antibodies in the semen of the person against his own sperm. So, anti-sperm antibodies which are present in the semen of the person. Now, these anti-sperm antibodies are of three types. They may be IgG, IgA, IgM. So, a person is producing antibody, the person's immune system is producing antibodies against his own sperm. So, this is called anti-sperm antibodies. Okay. Then thyroid dysfunction. Suppose a person has less than normal functioning thyroid, then his metabolic rate will be low, and this can also affect the hormonal levels in his body and it can affect spermatogenesis. impotency, inability to inseminate the female. Now the male is unable to achieve an erection, and therefore there is no successful insemination or ejaculation. Again, impotency can be considered to be a psychological factor. Then you know about the gonadotropins that play a very important role, that is FSH and LH. Sometimes the male may have a deficiency of FSH and LH. If FSH and LH are deficient, obviously the process of spermatogenesis will be affected. Then there are so many other medical conditions. Aspermia means complete lack of semen. There is no ejaculation, or semen is not ejaculated at all, isn't it? So aspermia, it may be due to a block. Say for example, a person has undergone a surgery in his prostate gland, or there is a growth in the prostate gland such as prostate cancer. Then there is no production of semen at all. This is called aspermia. Hypospermia is the person is producing semen but there is low volume of semen. Okay, normally the semen is about 5 ml of semen is produced at the time of about ejaculation. About 3.5 to 5 ml of semen is produced. If it is lesser in volume, then we call this condition as hypospermia. So remember, aspermia is lack of semen and hypospermia is low volume of semen. Then sometimes there is production of semen, I mean the seminal plasma is ejaculated, but there is absolutely no sperm in the semen. See the difference. Aspermia was lack of semen, but azoospermia, I am writing it here, there is lack of sperm. Semen is produced. I should not be calling it semen because there is no sperm in it. So there is only seminal plasma that is ejaculated by the male during coitus, but there is no sperm in it. So azoospermia, lack of sperm in the seminal plasma. Oligospermia is less number of sperm. So I'm going to call this as low sperm count. Normally the sperm count must be more than 15 million sperm per ml of semen or per ml of ejaculate that is produced, up to about 200. So it starts from 15 million to about 200 million sperm, that is per ml of ejaculate. But of course, if a person has less than 15 million sperm per ml of his ejaculate, then he belongs to a bracket of oligospermia, which is called low sperm count. Sometimes the sperm is not moving vigorously, so we call this condition as the lazy sperm condition. This lazy sperm is referred to as asthenospermia. That is, the sperm are produced, they may appear normal in their shape and size, but they are not moving vigorously, isn't it? So this is called asthenospermia. Then varicocele refers to within the scrotum, sometimes there may be dilation of veins. The veins in the scrotum may become broader and blood starts pooling inside. So pooling or collection of blood within the scrotum, because the veins in the scrotum, especially the veins which drain the blood from the testis, have become dilated. So you can imagine what will happen if there is collection of blood in the veins of the testis, as in the veins which drain the blood out of the testis. Then what happens is the temperature inside the scrotum will increase, and if the temperature of the scrotum increases, then obviously it will result in a condition called infertility, because spermatogenesis is drastically affected. Sometimes in male, the seminal plasma may also have very low fructose content. Fructose provides energy to the sperm, so the sperm do not have enough energy, and so again their motility may be affected, or sometimes there may be excessive amount of prostaglandin. Prostaglandins are hormones which bring about contraction of the female reproductive tract. They may bring about so much of a contraction that the egg may get washed away. So if there is no egg in the female reproductive tract, then you cannot there is no question of fertilization. So excessive levels of prostaglandins in the seminal plasma, and very low levels of the sperm energy source, that is fructose in the seminal plasma, can also be a cause for infertility in males. Now, infertility in females. So, what causes infertility? What are some of the causes for infertility in females? Sometimes the female may exhibit a normal menstrual cycle, but she is not ovulating at all. And this ovulation defect is referred to as anovulation. Then sometimes there may be a problem associated with, say, the luteal phase. It is called the luteal phase defect. That means something is wrong with the corpus luteum. Probably the progesterone is not being produced normally, or sometimes now here in an ovulation, probably there is something which is wrong with the LH. There is no LH surge. Do you remember when does the LH surge happen? It happens on the 14th day of the menstrual cycle. Okay, in luteal phase defect, again there are a lot of hormonal imbalances in the body that may affect the nature of the endometrium, and which may not allow proper implantation to happen, and therefore the female cannot become pregnant, isn't it? Then there are a lot of endometrial factors. Sometimes the endometrium develops elsewhere in the uterine wall. Normally the endometrium develops on the inner wall of the uterus. Sometimes in certain cases there is a medical condition that is called endometriosis. Now, in endometriosis, endometrium may be formed outside the uterus in the pelvic cavity. So, this is something which is very, very peculiar. Normally, where should the endometrium be? The endometrium should be in the inner wall of the uterus, but in this case, it is formed elsewhere. So, this condition is called endometriosis. Okay, then just like we saw, the male can have anti-sperm antibody in the semen. The female may also develop anti-sperm antibodies in the cervix, in the vaginal regions, and this is what may be a cause for infertility in female. It may destroy the sperm that makes its way inside the female reproductive tract. There may be a lot of defects in the uterus, defects in the fallopian tube, especially defects in the fimbriae. Imagine there is a funnel-like structure. You all know the funnel is called infundibulum, and it is thrown into finger-like structure called fimbriae, which is responsible for uptake of the secondary oocyte from the pelvic cavity after it reaches out from the ovary into the fallopian tube. So if there is something wrong with the fimbriae, then there is no uptake of egg, which can be a cause for infertility. Increased sperm phagocytes by macrophages. Normally macrophages or WBCs are found in the inner wall of the cervix, in the inner wall of the uterus, that is endometrium. Suppose the female is producing too many WBCs and too many phagocytes in her endometrium, then they may eat up the sperm by phagocytosis. If the sperm are eaten up, if the sperm are destroyed, there is no question of fertilization, and that's why the woman is incapable of producing a child, and therefore it is a cause for infertility. So what we have done here is we have summarized the causes of infertility in case of males and the causes of infertility in case of females. So by now you got a fair idea of what infertility could be due to. It could be due to defects in the male partner, or it could be due to defects in the female partner. Ultimately, infertility means there is no fertilization, or if there is fertilization, there is no implantation, there is no pregnancy, and the couple cannot bear, or they cannot produce children. Okay? What is the solution for infertility? This is called assisted reproductive technologies. Okay, it's actually technologies or techniques. It can be used interchangeably. So what are these assisted reproductive technologies? Now these are medical technologies or a recent line of treatment. Now these technologies can help couple who are unable to produce children to finally bear or produce children. It can help infertile couple to become parents. So we are now going to discuss about one of the major means by which we can overcome infertility, and this is referred to as assisted reproductive techniques. So you can see here, using these techniques, couples could be assisted in order to have children. Okay, so let us see how it is. What are the different techniques which come under assisted reproductive technology? The first and the most famous technology under assisted reproductive technology, you all, I'm sure, must have heard of it. It is called in vitro fertilization and embryo transfer. So IVFET stands for in vitro fertilization and embryo transfer. So what is this also called? Famously or popularly, it is known as the test tube baby technique. So you can imagine what is done in the test tube baby technique. First, the mother, that is, say for example, there is a couple, there is a husband and a wife who want to bear the children. So in the wife or the mother, there is stimulation of ovulation. They inject certain hormones which have FSH and LH like activity into the body of the mother, and the mother will now produce more than one. Usually she produces multiple eggs. Okay, so now what happens is the ovulation has taken place. Then they can retrieve or harvest the egg. Now this harvesting of the egg can be done with an equipment that is called laparoscope, by making a small incision in the abdomen of the woman. So it is not a very major procedure, it is a minor procedure. So remember, mostly a laparoscope is employed in order to retrieve or harvest the eggs by making an incision in the abdominal wall. Okay, so remember, first you should make sure that the mother or the woman who wants to become the mother is ovulating. So there you have to stimulate ovulation by administering certain hormones, and then once she has produced the eggs, by using this equipment called laparoscope, you will retrieve the eggs, and then you will place the eggs in a petri dish under laboratory conditions, isn't it? So that's why it is called in vitro. Vitrum means glass. So this fertilization does not happen inside the body of the woman. The fertilization happens in controlled or in lab conditions. That's why it is called in vitro fertilization. So imagine that there is a petri dish like this. So here the husband's sperm is also introduced. First the eggs which are collected from the woman are placed inside the petri dish, and then the semen of the husband is introduced into this petri dish. So you can imagine what will happen now. Fertilization takes place. Where is the fertilization taking place? Is it taking place in the fallopian tube of the mother? No. It is taking place in a glass dish, in a petri dish, which is in a lab condition. So it is not under natural conditions, it is lab condition. That's why it is called in vitro fertilization. Okay, so the eggs came from the donor woman, say for example the wife, and the sperm came from the donor man, say for example the husband, and both of them are potential parents. They want to bear children, they were unable to bear children by natural methods. That's why they are resorting to assisted reproductive technology. Okay, so the egg is placed in a petri dish, and the semen from the husband is introduced into the petri dish, and then obviously you will the process of fertilization takes place. And what is the outcome of this? The outcome is there is formation of zygote. Now what happens is, once the semen and the egg are introduced into the petri dish, they are placed in special incubators so that the conditions are similar to what we see inside the human body. The temperature is maintained, the ionic concentration, there is a watery medium, a nutrient medium that is provided here, which is maintained as per the concentrations of the body fluids in case of our bodies, so that it can entirely simulate the conditions which are inside the body. Okay, and the last step you cannot see here, so I'm writing here, it is called embryo transfer. So you get a zygote now. What to do with the zygote? Now there are two things we can do with the zygote. So we can classify the in vitro fertilization and embryo transfer into two more further techniques that we will be discussing here. One is called zygote intrafallopian transfer, that is ZIFT, and intrauterine transfer. So remember what ZIFT stands for: zygote intrafallopian transfer, and IUT stands for intrauterine transfer. So there are two follow-up methods for in vitro fertilization and embryo transfer. Is this clear to all of you how in vitro fertilization is carried out? Now I want you to think about certain things. Why it is called in vitro fertilization? Because fertilization happens outside the body, fertilization does not take place under natural living conditions. Fertilization takes place in lab condition in a petri dish. That's why it is called in vitro fertilization. And now for embryo transfer, there are two methods. One is called ZIFT and one is called IUT. So remember, the two variations of in vitro fertilization and embryo transfer are the zygote intrafallopian transfer and intrauterine transfer, which we are going to discuss now. Okay, so I hope it is clear. So here you can see a very good illustration of what is done during in vitro fertilization. So first, the female is you have to stimulate the woman who wishes to become the mother to produce egg. After that, you can see it's a very rough representation, but using a laparoscope, the harvesting of the egg is done, the collection of the egg is done, isn't it? And then the sperm from the husband is introduced inside. Here they have literally shown a test tube, but normally it's called test tube baby because of the lab condition thing. Normally it is done in a petri dish. So the egg from the woman is mixed with the sperm from the husband, that is in a nutrient medium within a glassware, where a zygote or an embryo is formed. And then later the embryo, in this illustration they have shown that the embryo is transplanted, is transferred into which part of the woman? That is the uterus of the same woman, isn't it? She wants to become the mother. So her own eggs are collected, her own eggs are introduced into a medium with her husband's sperm, and the zygote or the embryo, in this illustration they are focusing on the embryo. The embryo is introduced into the uterus of the mother of the woman who wants to bear the child. Okay. Now I told you there are two variations in vitro fertilization and embryo transfer. Now let us again recall what was the outcome of in vitro fertilization. The outcome of in vitro fertilization was we got the zygote. Now what you do to the zygote is what is depicted, what is shown over here, that is the zygote intrafallopian transfer. So zygote is the product of in vitro fertilization. So I'm writing it here. Product of in vitro fertilization is zygote. Now what is done to this zygote here? See here, the zygote, or sometimes the zygote that is present and that is developing in the petri dish, it is, so this is the zygote. Imagine it may be allowed to grow into a multicellular embryo which has less than 8 blastomeres or up to 8 cells in it. So 8 blastomeres. So either you take the zygote or you allow the zygote to develop into an embryo, a very early stage embryo which has up to how many blastomeres? 8 blastomeres. And what do you do to both of these? It's either the zygote or the early embryo which has up to 8, very important point, up to 8 blastomeres are transferred into the fallopian tube of the mother, of the to be mother. She's not a mother still, she will become a mother once the embryo is transplanted into her fallopian tube. So now you understood the meaning. It is called zygote because you either take the zygote or, yes, embryo up to 8 blastomeres. Intra, within, fallopian, that is fallopian tube, transfer. It is transferred into the fallopian tube. Did you understand? So this is a continuation of in vitro fertilization. What is the product of in vitro fertilization? Zygote. What do you do to the zygote? You take the zygote or you allow the zygote to develop into an embryo up to how many blastomeres? 8 blastomeres. So zygote or embryo up to 8 blastomeres. Where are they transferred? They are transferred into the fallopian tube of the female. Because it is transferred into the fallopian tube, what is this technology called? Zygote intrafallopian transfer. Did you understand very clearly? So isn't it a product of IVF? In IVF, we got a zygote. So you take the zygote or the embryo and transfer it into the fallopian tube. What is the criteria for you to transfer the embryo? It should have cells not more than or up to 8 blastomeres. Clear? See, it is shown here very clearly. Correct? So the fertilized embryo or zygote. So what is being introduced over here into the fallopian tube? This is the fallopian tube of the recipient. The woman who wants to become a mother, recipient woman. It may be the wife from whom the eggs were collected, or it may be another woman. Did you understand? So she is the recipient woman because she is receiving the zygote or the embryo. So please remember what is introduced into the fallopian tube. I'm writing it here for clarity again. You either directly introduce the zygote or you introduce an early embryo. How do you know it is early embryo? Because it has only up to 8 blastomeres. Alright, so this is called as zygote intrafallopian. See, it's very simple. If you just dissect the name, you will get the answer. Zygote intra within fallopian transfer. But it is not just zygote, or it is an early embryo up to 8 blastomeres. Clear? The third one is again a modification of IVF-ET. So what did we get in IVF-ET? I'm going to show you again. In vitro fertilization, we got a zygote, the product of in vitro fertilization. Now what you're going to do is, you are going to allow the zygote, which is sitting inside the petri dish, you're incubating it. So the zygote is inside. You are going to allow the zygote to develop into an embryo. Now this embryo has more than 8 blastomeres. See the difference. In ZIFT it was an embryo with less than 8 blastomeres. But here you are allowing the zygote to develop to an embryo, a stage of an embryo which has more than 8 blastomeres. And where do you put this embryo now? The embryo is transferred not into the fallopian tube. In ZIFT it was fallopian tube, but here it is transferred into the uterus of whom? Either the same woman from whom the egg was obtained or a recipient woman. Isn't it? A recipient woman is somebody who wants to become a parent. She herself cannot produce an egg, but she wants to bear the child. Isn't it? So this is what is referred to as intrauterine transfer. Why all this? Because naturally, when the husband and wife try to bear children, naturally it is not happening. As a result, we need all these medical interventions. You understood the difference? So the product of in vitro fertilization was a zygote. The zygote is allowed to develop into an embryo. Remember, I am talking about intrauterine transfer. Intra means intra, within, uterus transfer. So the zygote is developing into an embryo which has what? More than 8 blastomeres. The more than 8 blastomere embryo is transferred not into the fallopian tube, into the uterus. Did you understand? So two variations of IVF. One is zygote or embryo less than 8 blastomeres in case of ZIFT, or embryo more than 8 blastomeres in the case of IUT. Okay? In vivo fertilization. Now, what is in vivo fertilization? In which what happens is a mature egg. Now, for example, let us say that there is a couple. So, there is a husband and there is a wife. They are normal. But there is a couple who are infertile. They want to bear children. So, what happens is within this female, normal fertilization takes place following sexual intercourse. So fertilization happened inside the fertile female. Remember, this couple is normal. So what is the product of fertilization? The product of fertilization is the zygote or embryo. This embryo is retrieved from this woman. What does this woman now become? She becomes a donor because she is normal and she wants to donate her embryo. Where did the fertilization happen? It happened inside her body, isn't it? So we call this fertilization as in vivo. Now what does she do? She decides to donate her zygote or an embryo to an infertile couple who are unable to produce children. So she becomes what? This woman becomes a recipient. So couple 1 is fertile. Couple 2 have infertility issues. Did you understand? And the couple wants to bear a child. So where did the fertilization happen? Sexual intercourse and fertilization happens within the donor woman. And then the zygote or the embryo is transferred into the recipient woman. See? Subsequent transfer into an infertile recipient. Now this recipient woman is infertile. And this donor woman is fertile. The donor couple are fertile, but the recipient couple are infertile. Once again, why in vivo or in vivo? Because where did fertilization happen? In the donor female. So I must be calling her a fertile donor female. It did not happen in a petri dish. If it happened in a petri dish, you will call it as what? In vitro fertilization. Here, the donor couple took part in coitus, and inside the donor female, fertilization took place in her fallopian tube. That's why it is in vivo. But she doesn't want to be the mother. She can donate her embryo to an infertile female recipient. Isn't it? So this is referred to as in vivo fertilization. It happens in living conditions. Please remember, in vivo means in living, not in laboratory conditions, in living conditions. So this is the fourth in vitro, assisted reproductive technology from fertile couple to infertile couple. Gamete intrafallopian transfer. Now in gamete intrafallopian transfer, as the name, let's dissect the name. Gamete. See, it's very, very self-explanatory. I'm so sorry. So gamete, intra, into the fallopian tube. You're transferring the gamete into the fallopian tube. So you're transferring the egg collected from the donor into the fallopian tube of the other female. So there is a female and there is another female. Now this female is a fertile female. She can produce egg, but this female is an infertile female. She cannot produce egg. So what can be done? Now from this fertile female, the egg can be taken. Gamete, remember, egg is nothing but the Gamete. So the gamete or the egg harvested from the fertile female is transferred into where? Into the infertile female, into the fallopian tube of the infertile female. Now tell me, who is the donor of the egg and who is the recipient? The fertile female is the donor because she is donating her egg. The infertile female does not have her own egg in her fallopian tube. She has the donor's egg, so she becomes the recipient. Isn't it? And then once she has received the egg, the infertile female, may participate in coitus with her husband, and only the egg will be from another woman, and she will not be the genetic mother, obviously, because it is not her egg. So she will not be the biological mother, isn't it? Genetic mother, because it is not her egg. Did you understand what is happening here? So egg is collected from a fertile donor and transferred into the fallopian tube of the infertile donor. Now another thing is, this infertile donor, she cannot produce egg. That's why she wants a donor egg. She cannot produce egg, but the advantage is her reproductive organs are normal. Her uterus is normal. So what can she do? She can sustain pregnancy. There's absolutely no problem in her becoming pregnant and becoming a mother. Only thing is, she cannot produce an egg. See, it is suitable. The recipient infertile woman cannot produce an egg, but she can provide a suitable environment for fertilization and development. That means her fallopian tube is normal, her uterus is normal. Only thing is, her ovary cannot produce an egg. Did you understand? So fertile donor to infertile recipient, you are not transferring the zygote or the embryo, you are only transferring the egg. That's why it is called gamete intrafallopian transfer. And where are you transferring the egg? Into the fallopian tube of the infertile recipient. Clear? The sixth one of assisted reproductive technology is called intracytoplasmic sperm injection. This is a very micro manipulative procedure. See, this is the egg and the sperm is being directly injected into the cytoplasm. This is the cytoplasm of the egg. So using a very thin micro needle, see, the sperm is being injected into the female cytoplasm of the egg. Both the egg and the sperm are put in a petri dish, and under the microscope, the egg, as you can see, is held through suction by a macro pipette, and the sperm is being introduced through a micro needle. So you can imagine it requires a lot of sophisticated equipment and professional expertise. Expert doctors are required. So here, please remember, as the name indicates, intracytoplasmic. Intracytoplasmic means inside the cytoplasm of the egg. What is being injected? The sperm is being injected to bring forth what? Successful fertilization. See, the sperm is directly injected into the ovum. The ovum is held in place through suction, and a needle, a very fine needle, is pierced into the egg, and the sperm is delivered directly into the cytoplasm of the egg. Is that clear? Intracytoplasmic sperm injection. Artificial insemination. Now, when is artificial insemination carried out? It is carried out when the male partner cannot inseminate the female. He is unable to ejaculate or inseminate into the female reproductive tract. Or the male partner is suffering from low sperm count. Remember, oligospermia, less than 15 million sperm per ml of semen. So these are the two cases where artificial insemination becomes an option. Either the male is incapable of depositing the semen inside the female reproductive tract, or he is depositing the semen but the semen has low sperm count, oligospermia. In this technique, what is done is the semen that is collected from the husband or from the healthy donor, in case the husband is infertile and his sperm are incapable of fertilization, then the couple may also opt for a donor sperm, the donor semen from sperm bank. So the semen collected from the husband or from a healthy donor is artificially injected where? Either into the vagina of the woman or directly all the way up into the uterus. If the semen is deposited in the uterus, we call this technique as intrauterine insemination. See, artificial insemination is abbreviated as AI. Intrauterine insemination is abbreviated as IUI. Lot of 1 mark questions you have here. When do you call artificial insemination as IUI? If the semen from the husband itself or from a donor male, if it is deposited directly into the uterus, then it is called intrauterine insemination. If it is introduced into the vaginal canal, then it is simply called AI, artificial insemination. Insemination means introduction of the semen, deposition of the semen into the female reproductive tract. But here it is not happening during sexual intercourse or coitus, it is being done artificially. That's why it is called artificial insemination. Where is it artificially introduced? Either into the vagina or into the uterus of the woman. Clear? What are the limitations? So we learnt about so many different techniques of assisted reproductive technology. What are the limitations of assisted reproductive technology? Lot of high precision handling is required. Specialized expertise is required. Very expensive instrumentation is required, which is why it is not affordable by everybody. Only by the upper class, a particular section of the society can afford it, because it is very expensive. And then there are a lot of emotional, religious, ethical, social factors. A lot of people who believe that something artificial is against nature, isn't it? So it's like saying a couple has not conceived, they are not meant to conceive. So lot of religious, ethical and social factors, and they have resorted to some artificial means. There is still some stigma attached to it. So these are the factors which are limitations of our ART. Like I said, some people are of the belief that if they are not conceiving, then it is meant to be that. They are against the measures or some of the attempts towards conceiving. So all this kind of myths and misconceptions that create a lot of limitations for artificial assisted reproductive technology, especially in a country like ours. Okay? Alternatives for ART. The best alternative is adoption. There are so many orphaned and destitute children in our country who may not survive up to maturity if they are not taken care of. And there are also laws which permit legal adoption. And this is the best method if a couple cannot afford artificial or assisted reproductive technology. And let me tell you, assisted reproductive technology also don't have very high success rates. Even after paying so much money, and if the couple undergo the procedures, there are less success rates for each of those procedures. So considering all of that, the best alternative would be adoption. Okay? So what did we learn in today's class? That is the last lecture of the chapter Reproductive Health. We learnt about STIs, sexually transmitted infections. We learnt about assisted reproductive technology, IVF and the follow-up procedures ZIFT and IUT. In vivo fertilization, where fertilization happens within the body of a fertile female and the zygote or an embryo is donated to an infertile recipient female. Then we saw a woman can donate her egg to a woman who cannot produce one but who can become pregnant and bear a child, that is gamete intrafallopian transfer. Then we saw you can directly inject the sperm into the cytoplasm of the egg, ICSI. And then we saw artificially depositing the semen in the vagina or in the uterus, artificial insemination. Limitations of ART, and lastly we saw adoption as an alternative for ART. Homework question for today's lecture. Mention the early symptoms and long-term complications of STIs for 3 marks. Name one sexually transmitted infection which has no cure. List the 3 preventive measures for STIs for 3 marks. I had told you about the 3 principles one must follow to be free of sexually transmitted infections. 3-mark question. The last one is a very important 5-mark question. Any 5 assisted reproductive technology you have to explain in detail, which can be adopted by couple to overcome infertility. Is that clear? So this completes this 3rd lecture as well as the chapter of reproductive health. So until we meet next time, take care all of you and have a nice time. Thank you.